Hello, and welcome to Module 2 Biology, Section 3 on cell organelles. Moving on from the membrane, we are now going to discuss all the various components inside the cell that let cells do all their various jobs. Again, these foundational segments are meant to be a jumping off point just to get you clear on a lot of the terms, just some of the general sense of what a lot of these more systemic processes are referring to when we talk about, the real fundamentals. So, as far as cell organelles go, these composite type drawings are things I'm sure you've seen back in biology class. We got the plasma membrane on the outside, the mitochondria, which we know about, the rough and smooth endoplasmic reticulum, the Golgi vesicles, Golgi apparatus. A lot of these things are, the words that may certainly sound familiar, and, a lot of them are frequently invoked in writing. But today we're just going to go and review kind of what a lot of these things, a lot of these different organelles do and how they all kind of come together and why this is all relevant and important. So the first one we're going to discuss is the endoplasmic reticulum, which you can see there outside the nucleus. Now, the endoplasmic reticulum is divided into two subsections, really, the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Now, notice there in the diagram, most of the endoplasmic reticulum, or ER as I'm going to call it, because endoplasmic reticulum is a mouthful, looks like it's covered with little dots. Those are all the ribosomes on the ER, and they are absolutely critical for protein synthesis, as we'll see. The smooth endoplasmic reticulum is more where phospholipids, steroids, and other compounds are kind of processed and synthesized and released. Now, let's pop over to this next slide so we can kind of see those laid out. When people discuss the ER, I think one term that's often used is kind of like the internal highway of the cell. This is basically where RNA and different signaling molecules and protein encoding molecules come out of the nucleus. They're kind of made in the rough endoplasmic reticulum and then shifted and processed from the ER over to the Golgi apparatus and then kind of shunted out. So if we think of the endoplasmic reticulum and the Golgi apparatus, which we'll discuss later, as kind of the area where all of the products that come from the nucleus are kind of manufactured, processed, packed up, and transported. So the rough ER, again, with those ribosomes on it, hence why it looks rough or fuzzy, is where proteins are primarily made, and they're also tagged via glucose and via various side chains, which kind of tells the cell where to put them. Smooth endoplasmic reticulum, where phospholipids and steroids are made. This is also where calcium ions are stored, and we'll see why that is so critical when we discuss neurons later on. It's also responsible for processing and neutralizing of toxins. We can discuss that a little more at a later date. Now, as we stated, the Golgi apparatus receives, repackages proteins. This is basically when and we'll see this in the next diagram, when a protein is synthesized in the endoplasmic reticulum, the Golgi apparatus is where it's further tagged and kind of routed to its final destination. Lysosomes are the last thing we're going to discuss in this segment. And a lysosome, lyse means to divide or destroy, is where the cell actually contains destructive enzymes carefully packaged away in these little floating organelles of death for destroying cell components, or in fact to destroy the entire cell itself. And we'll talk about autophagy and what that means in just a moment as well, because I know that's a frequent buzzword, especially in things like intermittent fasting and ketosis. Here we go. Here is a diagram. You see the rough endoplasmic reticulum covered with all those little ribosomes, those little red dots. Proteins are being made. That little yellow peanut-looking thing is one of the proteins being made. Transported from the ER directly to the Golgi apparatus, where it is further processed, tagged, and sent off. In this case, it's being sent by a secretory vesicle to the surface, but it can also be routed elsewhere in the cell. Autophagy, which I've heard pronounced 30 different ways. Autophagy is the destruction of a cell's own structural components. Now, this can take place in multiple different ways, in multiple different manners. If we talk about macroautophagy, that is where a organelle, protein, something in the cell is enveloped by a separate kind of neutral membrane and partitioned off and transported to the lysosome and pulled into the lysosome for destruction. The microvariation of this process is when the component is pulled directly into the lysosome itself. The lysosome may even wrap around it. And chaperone-mediated is where specific proteins are recognized by a special tag. And this tag is very much protein-specific and transported into the lysosome for destruction. Now, the difference between the macrovariation and chaperone-mediated is that macroautophagy can just be any broken organelle can get pulled in by this special this packaging membrane, where chaperone-mediated is specifically a tag that's applied, and then the organelle is pulled directly in that way. Now, autophagy, we talk about it quite a bit when we talk about fasting, apoptosis, disease. There are certain dietary interventions, or lack of interventions, types of diets, etc., that are being touted as promoting autophagy. Now, why is autophagy important? Well, cellular structures aren't meant to survive forever. Cellular organelles can eventually wear out, and by that I mean that you can get, you can actually get damages to the organelles themselves. You can get even, protein encoding issues and errors for contractile proteins in muscle cells. These can get damaged, irreparably damaged in certain cases. A lot of these cellular structures are prone to eventual breakdown. As they start breaking down, the cell needs to scavenge these and basically destroy them to make room for new ones and to rebuild them. The other thing is also, if you think about it, when you say, well, do I break down muscle cells, because I need amino acids, when you're not eating enough, or, the process of weight loss, surviving starvation. Well, how do you think all those peptides get released? It's not like the muscle cells can just get, dumped into your stomach and reprocessed. It's this autophagy where the muscle cells, the contractile proteins, are actually pulled into lysosomes, and enzymes in there actually kind of chew them up and pull the amino acids back out into the composite parts, and then takes those amino acids and kind of brings them back into the pool that the body can then use. Now, as I said, as needed for growth and repair, a reduction in autophagy is when we talk about age-related degradation. We talk about age-related degradation of contractile tissue, various cell organelles of skin, etc. It is the reduction in this self-cleaning mechanism that is actually responsible for a great deal of diminished function in everything from organs to different processes in the body. So, seeing autophagy as a necessary process, yes, it's a destructive one, but it is also absolutely crucial to the health and well-being of every cell in our body. As we can see, cancer, tumors, the uncontrolled proliferation of cells, whether they be undifferentiated cells or more terminally differentiated cells. In other words, are they just random satellite cells, or are they individual types of tissue? Cancer, you think, would benefit from both suppressing this process and promoting it. Because certain cancer, if you suppress this process entirely and a cell is just allowed to grow and proliferate, even with all these poorly encoded proteins and everything else, you'd think that by suppressing this kind of self-cleaning process, cancer grows uncontrollably, and that is indeed true. However, it also promotes it in certain other ways, and that's getting into a larger discussion of cancer and how promoting this process actually allows certain cancer cells to actually basically fight off destruction from external cells. But we'll talk about that later on. Something else, we talk about neurodegenerative disease. We talk about what they call these amyloid plaques, etc. We talk about neurodegeneration. A lot of these bad proteins or poorly formed proteins, which are actually, as we'll see, the creation of other types of cells, or glial cells. These proteins are not adequately tagged and destroyed, so it could be a reduction in this autophagy that actually kind of extends and allows neurodegenerative diseases of certain sorts to take place. Of course, the interesting thing is not all autophagy is created equal. It's neither good nor bad. For example, let's say, if we're talking about this process and we're saying, okay, autophagy is associated, reduction in it is associated with aging and with certain kinds of cancer, etc. So let's do what we can to really enhance and optimize this self-cleaning process. And we know that fasting induces autophagy, and so therefore, fasting can fight cancer. And again, this is that kind of syllogistic argument that really doesn't make sense because autophagy caused by fasting, for example, it's a certain basically pathway. ATG7 is one of these pathways where starvation induces a certain kind of autophagy by basically scavenging certain types of proteins on a preferential basis, kind of an as-needed basis, reducing the amount of unneeded structures, et cetera, to allow the body to survive in a lower calorie environment. If we talk about age-related degeneration or cancer-mediated reduction in autophagy, simply promoting one particular kind of autophagy does not necessarily mean we're going to randomly happen to promote the kind of autophagy needed to, say, halt tumor production or reduce the aging process or reduce neurodegenerative diseases, etc. Autophagy is not an on or off switch, depending on whether it's the macro autophagy or micro or chaperone mediated, depending on whether it's caused by exercise or it's induced by starvation or fasting or whatever. The type of autophagy that takes place very much depends on the stimulus. So simply inducing the body to start to kind of upregulate this self-cleaning process does not mean that you are going to upregulate it in the ways that may be needed to treat a certain disease. It's basically like saying, you work in a factory or something, and the factory's getting messy and it's getting dirty and there's stuff everywhere. So let's have a cleaning crew come in, but the cleaning crew is only going to work in one room, or it's only going to clean one machine. That's not going to fix all your problems. The likelihood that if the factory is messy and one machine isn't working right and you don't know what machine it is, having a single crew come in and clean one machine somewhere is not necessarily going to guarantee you're cleaning the right machine. The body is simply too complicated. So there's no definitive link, as it says in the end there, that selective autophagy can reverse a disease process or slow any disease process or aging or anything else. Certainly when we talk about a lot of fasting or calorie reduction for longevity, we're not really talking about promotion of this process. We're more talking about like lower levels of free radicals, lower body mass index, etc., and other factors associated with longevity. So if you see that argument being made out there, just know that it's probably a little premature and doesn't really know how this process works. So there we go. That's lysosomes and autophagy. Next, the mitochondria. Singular is mitochondrion. So this meme I had to include simply because I like them, because we've all learned this in school. Of course, the mitochondria are not the only powerhouses of the cell. Certain processes take place outside them, like fermentation, but we won't discuss that just yet. What do the mitochondria do? Well, fun facts about mitochondria: they do in fact have their own DNA. And in fact, tracking mitochondrial DNA is a really good way to establish, is actually one of the things that's looked at in everything ranging from disease progression to maternal bloodlines. Its own fascinating course of study. Anyway, they have their own DNA. The basic sense is that mitochondria used to be their own basically a free-floating sort of bacteria that eventually was incorporated into some of the first eukaryotes, and because it was a very much a commensal relationship between the two, and now they are integral to cell function. What do they do? Well, they are where cellular respiration takes place. And we'll talk about that, of course, when we talk about energy systems. But all you really need to know now is that when we talk about the Krebs cycle and cellular respiration, all of it takes place pretty much there, and we'll see how all of that works. So they are, in fact, mostly the powerhouse of the cell. They're not where the fun process takes place, though, which is fermentation. We're not, unfortunately, talking about beer. We are talking about lactate or lactic acid fermentation. That takes place in the cytosol. which is why certain cells that lack mitochondria, like red blood cells, utilize lactate fermentation as their primary energy source. Now, the number of mitochondria very much depend on cell function. Obviously, skeletal muscle would have quite a few, whereas, again, like red blood cells have none, and certain less energetic cells that don't have as high an ATP need may have even lower amounts. Now, to actually proliferate and grow or increase the number of mitochondria and increase the number of mitochondrial enzymes, this is a fairly involved process. This regulator called PGC1 alpha. Don't worry, none of this is going to be on any exam. All you need to know is that there are a few different things that can activate PGC1, including, interestingly enough, nitric oxide on its own, which may be a particularly interesting discussion later on external compounds and pathways that can increase endurance. Because remember, mitochondria produce energy. You increase the number of mitochondria, you increase the amount of ATP that a given cell can produce. Now, AMP kinase, AMPK, we've heard about this in exercise and how aerobic exercise triggers AMP kinase, which can increase mitochondria number. How does that happen? Well, ATP ordinarily blocks AMP kinase activation. Now, adenosine triphosphate is the adenosine base and 3 high-energy phosphate molecules attached to it. ATP has 3. ADP has 2, AMP has 1, and these are in order from the most energy potential to the least energy potential. Now, in the cell, there is normally a 10-to-1 ratio of ATP to ADP. As that ratio increases, this can actually activate and change a number of things. As the ratio decreases, it can activate other things. Remember in the discussion on cell membranes and channel proteins and insulin, a rise in this ratio in beta cells in the pancreas is what stimulates insulin release. In other cells, as the amount of ADP and AMP increases, and I see, look, energy stress, what's going to happen is as the amount of ATP reduces, what's going to happen is that AMP, which is the lowest energy version, is going to increase as well because ATP is reducing. So now, all of a sudden, there's more ADP, which means occasionally in pathways, ADP is going to be converted to AMP when energy needs get very, very desperate. Remember, ATP is the biggest source of energy. ADP is mostly a byproduct, but in a pinch, it can also dissociate phosphate. AMP is the rock bottom source of energy. If a cell starts to get hypoxic, ADP reduces or ATP reduces, and then suddenly even ADP will occasionally lose a phosphate, so you start getting AMP. AMP kinase then activates. Now, ATP also blocks the activation of AMP kinase. So as the cell burns through energy, suddenly there's less ATP to block the activation of this, and adenosine monophosphate starts activating it. So basically, AMP kinase is activated when a cell gets into an extraordinarily low energy situation, basically when the gas tank is flashing empty. It is activated by hypoxia, hypoglycemia, muscular contraction. If you think about it, all three of those would definitely instigate this. Now, why is that? Hypoxia. Well, hypoxia, aerobic ATP generation is not taking place, so you're left with fermentation. And what's going to happen is fermentation is extraordinarily inefficient. And if a cell is in a hypoxic state and really going through heavy fermentation, what's going to happen is that ATP is probably requiring ATP at a faster rate than fermentation can generate it. So any exercising muscle that starts to go hypoxic and is still undergoing heavy contraction is going to start activating AMP kinase. What's going to happen is AMP kinase is then going to trigger this downstream process that's going to be responsible for the synthesis of more mitochondria, which makes sense. Again, endurance exercise, more mitochondria, because the body is saying, oh, there's not enough energy to keep these, to support all these cellular processes the way the body is currently, status that the body is currently at. Yep, there's the mitochondria. The nucleus. Nucleus contains all of the cell DNA. Now you can see the endoplasmic reticulum around it with the cisternae. That's a fun word. This is basically, every cell has a different looking nucleus, but it's always very easy to spot. You can see all the chromatin, all the chromosomes on the inside. We know what the nucleus does at this point. Now, nucleus is what contains the cellular DNA. That will be a separate discussion on transcription and translation. Most cells, now the red blood cell, again, just as the kind of, redheaded stepchild of the body, has no nucleus either. Most cells, including smooth and cardiac muscle, have one nucleus. Skeletal muscle and osteoclasts are very weird cells, by the way. Skeletal muscle has multiple nuclei. Now, why do skeletal muscles have more than one? This is actually a topic that is under heavy discussion right now. We in fact may be changing some of these ideas as we learn more. But effectively, as you can see in item 1, multiple nuclei allow for more protein synthesis to take place. The current theory, which is still, again, subject to change, is that a single nucleus can only really be utilized by a certain cell volume to carry out cellular processes to transcribe sufficient proteins. And if you think about it, this makes sense because a single nucleus contains one copy of the host cell's DNA. And all of the various processes that take place within a cell are going to need to get to that DNA to effectively carry out protein creation, protein translation. If the cell gets way too big, and if we say we have a very big muscle cell with so many contractile proteins, now remember, we got contractile proteins, so we got autophagy breaking down these proteins, we've got new ones being made, we've got all these huge energy requirements, and we've got all these enzymes that need to be made to transport everything, and all these various hormones coming in and processing and transcribing. Eventually, if you think about it, that nucleus is going to get overwhelmed, and skeletal muscle is one of these things in the human body that is just wildly, ridiculously capable of changing size to adapt to the body's need. So what happens in the human body, well, in most organisms, is that as a muscle gets the signal to start growing, these satellite cells surrounding the muscle effectively are differentiated. Now, when we talk about satellite cell differentiation, satellite cell is basically a germ cell that can become anything. As it starts to differentiate, it means certain processes start to activate that make the cell take on a unique characteristic. These satellite cells take on the characteristics of muscle cells, become myoblasts that are then incorporated into the existing muscle cell itself. That nucleus then becomes part of the main muscle cell. As you get more and more nuclei in the muscle cell, you have more sites available for simultaneous transcription, translation, and therefore the muscle's ability to respond to demands increases dramatically. Now, satellite cells are increased when you're younger, anywhere up to about the ages of 40 to 50, by exercise. The rate of satellite cell proliferation very much declines with age, and past a certain age, in fact, you no longer get much satellite cell differentiation at all. So this is why actually building muscle when you're younger is much, much easier than building muscle when you're older. In fact, if you see individuals in their 60s, 70s, 80s with significant hypertrophy, even with the use of anabolics, chances are those people trained significantly when they were younger. And I don't want to say this definitively because I'm sure something will come out that disproves it eventually, but if you see an individual who is older, who is training even while using various performance-enhancing drugs, etc., the possibility that they will build significant muscle past a certain age is very, very slim. They can increase it to a certain extent, but unless they have that myonuclei proliferation from an earlier age and that satellite cell differentiation, they probably won't be able to significantly, hugely hypertrophy their muscles. Anyway, that's neither here nor there. The very interesting things about myonuclei is that they resist autophagy and apoptotic, which means self-destructive, behavior on disuse. If you are an individual with a significant amount of muscle mass and you stop training for about a year, now assuming you are not going into huge caloric deficits, which will just lop off slabs of muscle. Say you lose even a significant amount of muscle over the course of that year. Even though the muscle size has reduced, the amount of nuclei in the cell are very, very, very resistant to destruction. And that makes sense, that the cells don't have too many pathways for quick nucleus destruction, especially a healthy nucleus. In this case, there's various research that's showing various rates, and this is all still very much current, so we may have to update this portion in a couple years. But the fact that the myonuclei can resist autophagy as muscle cell is being actively catabolized actually has profound implications on certain things that we may see, including doping bans, for example. This has even become kind of a hot topic in the whole, the whole concept of male to female trans athletes. If an individual, through the use of performance-enhancing drugs, or even through, say, difference between men growing up versus women, have a significant amount of skeletal muscle built and a significant amount of myonuclei proliferation, as you'll see in tremendously hypertrophied muscle, even if they then cease the utilization of performance-enhancing drugs, let's go back to that in doping bans, the new myonuclei which were synthesized because of this favorable anabolic environment may be much, much higher than a natural athlete could attain at that point. If those individuals then lose the additional muscle when they are no longer taking performance-enhancing drugs, they still carry those additional myonuclei which make their existing muscle more responsive and potentially, I say potentially even though it's a high probability, potentially more quick to respond to hypercaloric environments, which means it's easier to build muscle for those individuals, in a mass gain phase. They are, potentially able to respond faster to various metabolic demands, which means even for endurance sport, they may be able to synthesize a lot of the enzymes needed for contraction repair, everything else much faster. So these more heavily myonucleated muscles may still convey a performance advantage. Again, I say may. I don't want to say they definitively do, because, again, nothing is really definitive in this field just yet. But this is why, to a certain extent, doping bans can last for a number of years. How quickly do myonuclei eventually go away? Well, the tricky thing here is it really depends on what tissue you're looking at. If you look at tissue like the calves, and you would use calf muscles as a basis for determining myonuclei, how long they stay around for. In calf muscles, they may only stick around for another 6 to 12 months. So after 12 months, your performance-enhanced, more heavily myonucleated calves may go actually back down to kind of a baseline, especially if you're in a hypocaloric state. The trapezius muscles and some of the muscles up around the shoulders, however, which people typically associate with being quote-unquote more responsive to anabolics and training, the myonuclei, the additional ones there, may linger for more than 6 years. Now, all of these have been done by looking at biopsies from different types of athletes over the years. So you can make a case for a shorter doping ban by using calf biopsies, or make a case for an almost lifetime ban, if you would like, by looking at trapezius biopsies. So this is why a lot of the science, when it comes to muscle memory, for example, muscle memory is typically attributed to presence of greater number of myonuclei in previously trained muscle. But a lot of the debate about muscle memory and doping bans, and even talking about any quote-unquote advantages held by male to female trans athletes, a lot of it is very subjective and has to do with what tissue is being examined. So that's a little bit about multiple nuclei and skeletal muscle. This is one reason why we kind of say that weight training is very important for all people at various phases during their youth, especially because we talk about the satellite cell reduction in satellite cells over time. Important to note that both aerobic and resistance exercise can cause myonuclear proliferation. They can also increase the number of satellite cells, increase the size and proliferation of the endoplasmic reticulum. So remember, aerobic exercise does this as well. So even aerobically trained athletes when they're younger may be able to build more muscle later in life, which is interesting. So Interesting implications there for resisting age-related muscle loss as well, or disease-related muscle loss. So if you want to check out further reading, I would look at your OpenStax Anatomy and Physiology, both sections on organelles, but there's also the sections on protein synthesis, pages 110 through 115 and 121 through 125 on stem cells, may give a little more information on a couple topics we've discussed.